The overall goal of this procedure is to help users establish a high-throughput collection and processing pipeline of cryo-electron tomography.
This method can help answer key questions in the cryo-electron tomography field, such as how do I handle data collected with new generation detectors and process the huge amount of data produced by automated tilt series collection software. The main advantage with this technique is that the user can configure multiple software packages in a unified manner, defining an efficient pipeline for automated tilt series processing, allowing for more time to be spent on tomogram analysis.
Generally, individuals new to cryo-ET will struggle because many tedious steps are required to handle massive data generated from the electron microscope. We first had the idea for this development 5 years ago when we had to manually go through hundreds of steps to get a 3D reconstruction of one bacteria.
In advance, consult the text protocol on preparing the Shigella flexneri minicells. Then, prepare the electron microscopy grids as explained by an existing JoVE publication and also documented in the text protocol. After having loaded the EM stage with a collection of minicells embedded in vitrified ice, start collecting low magnification maps for the tilt series. While viewing the fluorescent screen at low magnification of about 2300x, find grid squares that are acceptable to image. This location should contain thin ice and the subject of interest with no contaminants. Next, adjust the view to the center of the grid square.